Thank you all for being here. It's my pleasure today to talk about some of my recent work. So I'm going to talk today about RNA-Seq data. So I don't think it's data that that a ton of people here have been talking about so far, but from my experience, it's something that people are using a lot in the lab. So hopefully some of the things I'm going to talk about are going to be relevant for you or collaborators or whatnot at some point. The lion's share of this work was done by Kieran Evans, who was an undergraduate with me at Pomona College a few years ago, and he's now a PhD student at Carnegie Mellon. And then it's also worked with our collaborator, Dan Stovall. The impetus was working on his data that is on E. coli, but I don't think there's really any E. coli data. Okay, so just kind of really going to the basics of what RNA-Seq data, how they're collected and what happens. We've just got, this central dogma, and the gist of it is, at the end of the day, we get reads that are aligned to a reference genome and counted. Okay, so that's like the basics of what you need to know about RNA-Seq data, is that the data you get are a bunch of counts for each gene that is in this reference genome. And we want to be able to compare the genes across two conditions. So I'm going to be talking about normalization, and I'm going to be doing it within the context of this two-sample comparison, condition A and condition B. But really the things I'm talking about are relevant for whatever sort of experimental design you have and whatever kind of research questions. But just to sort of make it easier, it's more straightforward to just talk about this two-sample comparison, condition A versus condition B. And we're interested in for a particular gene, what's happening, and at the end of the day, are there more reads being mapped for that particular condition? There's lots of ways to define a transcriptome, so that's just up to you. You can talk about all the RNA in your cell, in your experiment. You can talk about only protein coding RNA. If you're interested in a particular subset of RNA, then that's again up to you, up to your experiment. But then you have to ask your question about what's your biological question of interest. Are you interested in changes in gene expression per cell, or are you interested in gene expression per transcriptome? And we're really going to focus on this first one, though. About halfway through the talk, I'm going to give you an example of an experiment that was done where there was just a real sharp decrease in gene regulation overall. So there was just much less RNA in the cell, and so then the researchers decided that they were interested in that second bullet point. Okay, so but for us, for right now at least, we're going to be talking about a gene being differentially expressed across conditions A and B. If there's a difference in the amount of RNA per cell produced under different conditions. So when I say per cell, I mean like at the top of the diagram, right? Not at the bottom of the diagram. And so how we go through this differential expression analysis is that we measure this gene expression under each of these conditions. And that's happening at the bottom of the diagram, measuring those reads. But then we have to normalize, and we're going to spend most of this talk talking about that normalization, the gene expression, so that we have an apples-to-apples comparison. And then we can do hypothesis testing for differential expression. And I'm not really going to talk a lot about differential expression today, just a tiny little bit. And again, your third step might be something totally different. So I like this quote. It's from a paper that's now about 10 years old. It says, the greatest impact on differential expression detection is the choice of normalization procedure. So these guys are not the only ones to have found that normalization really is the thing that's driving a lot of outcome. Okay, so again, here's this picture, and we're really wanting to know how much expression is happening at the cell level, but how many reads we get, which we're going to call the library size. The library size is going to be the total number of little bars we have down here, the total number of reads. Well, that depends on lots of things. For example, it can be spent. It can be determined by the amount of time that the sample is spent in the sequencer, right? So if you just let the sequencer keep sequencing, you'll get more reads, and that's going to be a source of technical variability. There's the mRNA expression, which is the thing we want, right? That's how much how much mRNA there is in the cell overall, which is the thing we're trying to measure. That's the biological variability, and that may change that hopefully will change from condition A to condition B, because that's the whole reason we're doing this experiment, right? That's what we want to see, and that's what we want to find. Of course, there's lots of other factors. Some of you may be familiar with, working with this type of data. There's, the gene length, right? The longer the gene, the more reads are going to just kind of naturally fall in that gene. The amount of GC content determines also the number of reads that we get. Okay, so library size is the total number of counts you get at the end. And the goal is, again, to compare in an apples-to-apples way the number of reads per gene to see if the expression has changed, but keeping in mind the library size, keeping in mind what happened on that previous slide with the different aspects of variability. Okay. So here's something that happens. Sometimes in experiments, there are one or a few genes that drive the expression. So in this first bullet point, they observed that in one of their samples, over 50% of all of their reads came from a single gene. In the second example, 50% of the reads were concentrated in about 5% of the genes. And in the third, 50% of the reads were in 45 genes for the male flies and 186 genes for female flies. So now let's talk about what happens with this situation when you end up with one or a handful of genes that are really dominating the library size, right? They're totally determining the overall number of counts in the final data set. So this is a picture that I'm going to show quite a few times. So different varieties of this picture. I'm not just going to show. So I'm going to spend some time talking about what each piece is. So we're going to start all the way on the left with that A, and that's mRNA per cell. So that A on the very left, that's truth. That's what we want to get out of the experiment. That's what we're trying to figure out. And what we see is that in condition A and condition B, the blue and the green genes, the ones on the left, are not differentially expressed, because that's how we're defining differential expression, is the amount of mRNA per cell, right? We're not defining it as the number of reads at the bottom. We're trying to get back to the top of the diagram, how much mRNA is being produced in the cell. And it turns out that there's this one gene that has a tremendous impact, and it's that purple gene. And in condition B, it's has twice as much mRNA per cell. And so what happens when you move to figure B there is you see that the green, the blue, and the purple all have about 1/3 of the total library size. But in condition B, the purple has half of the library size, and that the blue and the green have a quarter. Okay, so if we just count sort of the number of reads at the end, what we end up with is, so these are the reads that get aligned, and we end up with a picture like this that says that the blue and the green are down-regulated, and that the purple is up-regulated. But this is not actually what happened, right? And this is what would happen if we did something like total count normalization as well. So total count normalization, just an RPKM, very similar, says if you take the total number of the total number of reads that we get right here, and just sort of divide by everything, then we end up with this sort of ratio, those bars, and we end up misleading ourselves by thinking that that the blue and the green are, oh sorry, I put A on top here. So that the blue and the green would be upregulated in condition A, and that the purple would be downregulated in condition. So this node here is not trying to be flippant. It's just trying to say, like, you want to see changes, right? You want, when I say this worthwhile experiment idea, like the whole point is to really kind of make things change. And so if they're changing in ways in particular like this, that you're going to get problems. I mean, I've obviously made this example quite extreme here, but you're going to get problems like this when you just use total count or RPKM for your normalization technique. So RPKM, I think I had so RPKM was, 2008 was the reference there. So that's like, the original sort of apples to apples. Let's make sure we're not, comparing how long it was in the sequencer. But since then, there's been lots of other efforts to think about different normalization techniques. Okay, so I'm gonna not go through every single one of these. I'm only gonna go through 3 classes. But the first class here is this sort of overall count, and the underlying assumption there is that in the 2 sets of expressions or 2 sets of conditions, they have the same total expression. So the amount of total expression is the same under the different experimental conditions. So that's, I'm not actually going to leave you with much truth. So apologies for that if you were expecting truth. You got to go to the philosophy department to get that. But I'm going to just leave you with, here's the assumption that's being made if you're going to use this method. That's sort of what this talk is. Here's the assumption. If you believe that assumption, then use that method. Okay. So here's another example where a handful of people were all studying sort of aging in yeast, and what they found was that roughly equal numbers of genes are up and down regulated in response to aging. But then Professor came along and he found a substantial loss of histone proteins from the genome as the cells age in yeast, and it increased the transcriptional activity of the genes. And he was able to use spike-in controls to find that we don't indeed get an equal proportion of up and down regulated genes, that indeed there is some global regulation happening as cells age on a per gene basis. So what is that? What's going on here? Okay, so the truth in part A there is that we see global upregulation. So we see that the mRNA is what we want to be. There is more mRNA in condition B. But then we end up sequencing the same number of molecules, and we get this situation where whatever we normalize with, we just can't differentiate these two, because we have, again, this idea of just where the technical variability, we can't distinguish the technical variability from the biological variability that's happening here. And so we're going to observe that there's a fold change of 1, when indeed we want to be seeing a fold change of 1/2. So what the researchers did was they added a control gene. So a control gene is a gene that is not impacted by the experimental conditions, so condition A, condition B, but does hopefully, fingers crossed with control genes, does react to whatever the technical aspects are in a way that the other genes are. So if you leave it in the sequencer longer, that it'll keep sequencing and whatnot. So sometimes it's tricky to get control genes to behave exactly the same way as the rest of the organism.
So, like, what kind of gene would this be? Like a house?
Yeah, like a housekeeping gene, exactly. So I forgot to preface my talk by saying I'm a statistician. So sometimes it's housekeeping genes where we actually kind of know behavior and it's genes involved in, whatever, cell death or something. And sometimes it's spike-in controls. So again, as a non-biologist, I'm not totally sure which experiments can do these and which can't. But exactly, so either housekeeping genes or spiking controls allow us to say, okay, I'm going to normalize by making sure that the overall expression in that housekeeping gene, which on the slide here is purple, will be the same across these conditions. And in this case, then we see that we're able to determine that there is an upregulation in B, a downregulation in A. Like that should be switched. Okay. So there we go. So there's a whole class of normalizing techniques that use housekeeping genes, spiking controls, and what are they assuming? Well, the controls have to exist, and they have to behave as expected, which means this behavior as expected means they can't change based on the experiment, right? The condition A, condition B, they have to be negative controls, like they are not changing. But then this, my understanding, is even a bigger issue, right? So especially if you're doing a spike in experiment. So you have to get the technical effects to be the right ones that are sort of the same for the spiking controls as for the rest of the genes in the sample. Okay, so these researchers found that there was downregulation for every gene, and indeed that there was like this 30 to 50-fold decrease of the total mRNA level, and they decided that they wanted to measure on relative expression. So they actually, again, back to this, like, what do you want to know? What are you hoping to measure? They said, okay, we're clear, we are clear, and you, dear readers, you're clear that we have this down, this expression that's changing, but what we're curious about is how it's changing differentially, right? And so go for it. Do it. Yeah, that makes sense, right? So like for all the genes that were, 1/10 of the original mRNA per cell, now they're, 1/5 of the other one, right? So it's maybe not interesting in this case to say that everything is downregulated because we already know that it's downregulated, right? So again, just kind of thinking about what it is that we want and what it is that that we're measuring. Okay, so the essential normalization idea is that that the numerical value, whatever that is, right, whether it's counts per mRNA per cell somehow or per the transcriptome or whatever, but the ones that are not differentially expressed should not vary as a function of library size or biological condition, right? These are the non-differentially expressed ones. That's what I mean when I say non-differentially expressed. I'm saying the biological, they don't change across the biological condition, and they also don't change as a function of the library size, which includes technical variability and whatnot. And so how do we do what we were doing in those pictures? Well, what we do is we work with something called a size factor. So a size factor is a number that's typically sample specific, though it could be gene and sample specific. But in this talk, we're just going to be talking about sample specific, such that the values of the non-differentially expressed genes do not vary as a function of these things. So for example, in this one, we just divided everything by the value of the purple gene, right? Because we were forcing the purple gene to be the same across the two conditions. And so then we said, okay, that's right, because in this condition B, these were all in the sequencer together, right? So these are all genes that live in, they're on the same sample. Of course, this gets slightly more complicated when we have lots of samples and stuff, but it's not really any different. You just end up taking averages and stuff. So we divide by a sample-specific size factor in order to make sure that the non-differentially expressed genes do not vary. Okay. So I have one more class of normalization techniques to talk about, but before I can get to that, I want to talk about some of the assumptions that we've already addressed, just so that it's easier for me to talk about that third case. So here's a situation where where both samples have the same mRNA per cell. Okay? And we see that out of the 6 samples, a third are upregulated and a third are downregulated. Can we see that? So this is some kind of measure of the mRNA amount. And so you add up all these bars, and when you add up all the bars, you see that there's the same mRNA in the condition A and as in condition B. And then you can see that the first 2 genes are upregulated in condition A, and the last 2 genes are downregulated in condition A. Okay? So this is helping us differentiate the idea of the number of genes, which are up and down, and that's where we're going to get to symmetry. Symmetry is going to be the number of genes that are upregulated versus downregulated, and the amount of mRNA per cell is the amount. So just to kind of make that more clear, here is symmetric, right? 2 are upregulated, 2 are not differentially expressed, and 2 are downregulated. So that's symmetric, but there's a different amount of mRNA per cell at 4 of these plots. So here we have the same mRNA per cell, but we have 3 that are upregulated, 2 that are not differentially expressed, and 1 that is downregulated. So we have asymmetric, right, because the number of upregulated genes is 3, the number of downregulated genes is 1, but we have the same mRNA per cell, and the last one, you knew I was going to say this, right, 3 and 1, but they have a different amount of mRNA per cell. Okay, so now you guys got that. Let's talk about this last type of normalization. So this class of normalizations takes the genes and thinks about sort of the median expression in the thought that, even if I don't have equally the same number of genes that are upregulated and downregulated, that median gene is probably not up or downregulated, right? Because I need more than 50% of my genes to be upregulated or more than 50% of my genes to be downregulated if the median gene was not. So there we go. Pretty straightforward. Each read count is divided. We said we need this size factor. Here's the size factor. Size factor is always going to be sample specific. Is divided by the median read count in the sample. So we just take the sample's median and we divide by that value. And in this case, it doesn't really matter in condition A. In condition B, it's either of the green or the blue, and we end up with the correct normalization. So now we're going to talk about DE-seq, which is arguably, the most common normalization procedure, and really builds on that idea of the median value. So what DE-seq does is they take not only the median value, but they use it's almost like a reference sample. So let's see what these numbers all are. Kij is the number of reads aligned to I under sample J. And so we're going to try to find for sample J, what is that size factor? What is that number that we're going to be dividing by to make sure we get this apples to apples comparison? Okay, so the size factor, so this is not DE-seq yet. We're going to get there, but we're going to pretend it is for just, 2 slides, I think. So the size factor is if we could find all the genes that are not differentially expressed, okay, so that's what I'm saying here. So gene I is not differentially expressed across these samples. We would take the median of those values, and we would sort of have this relative expression value that they're all going to be kind of scaled by the same thing, and it's just a way of making it a little bit more numerically stable. The numbers come out a little bit better. So let's see what happens in practice. Okay, so we have these samples, and we have green and purple are not differentially expressed. Okay, so the green is the first one, the purple, I know the colors sometimes kind of seem wonky. So the first one and the last one are not differentially expressed. And so what do we have here? I just made up some numbers. Well, I didn't really make them up because I wanted them to work out nicely. So I used, fake numbers. I mean, they're actual numbers. 24 is a number. It's not like it's not a number, right? Okay, so anyway, so I have to do this thing that it told me to do. So I take the median of the counts, Kij, divided by this reference sample idea, and I get my size factor for sample A, condition A is √2, and my size factor for condition B is 1 over √2, and look, my size factors. Oh, wait, look, hey, A is twice as big as B. Right? See, A divided by the size factor of A divided by the size factor of B, √2 over 1 over √2 is just 2. And that's what we want, because you can see here that the green is these non-differentially expressed. It just happened to be that condition A, let's say, was left in the sequencer twice as long. Okay, so this is the gist of DC. But in the vast majority of cases, the median of the non-differentially differentially expressed read counts actually equals the median of all the read counts. So the only case where that first sentence is violated again is when you get into situations where you have huge proportions of differentially expressed genes, right? Again, if you have, huge proportions of upregulated genes or downregulated genes, right? If you have more than 50% up or more than 50% down, then that first one is going to be violated. But anyway, this is DC. It's you find the median. So we're making that sentence at the top right there is this equality here, just that they're wanting to find the median of the non-differentially expressed genes, and they're just using the median of all the genes, which is a reasonable thing. Okay, so this is DC. There's also, other ways of trying to get around the differentially expressed genes. So this is an algorithm step to figure out which of the differentially expressed genes, then find the median, then test again, find the median, test again, right? So like kind of going back and forth with normalization and differentially expressed genes, so that we can actually get at this, right? We can figure out which ones are the non-differentially expressed genes. Because I don't know if that was clear, that we haven't done differential expression when we're doing normalization, so we don't know this, right? That's why we had to make that assumption here, because we didn't know which of the genes were differentially expressed, and so we said that we were going to just use all of them. But we could go back and forth across those two, and that's a normalization called DEGES. This is a different normalization technique. Okay, so this is this class of all of these genes. They are assuming that the differentially expressed and the non-differentially expressed genes behave the same in terms of the technical effects, and that there's roughly symmetric differential expression across conditions. And you've probably heard of and or used many of these. So this is just sort of how I have classed these different normalization techniques. I'm going to now move on to some simulations that we did, but a lot of them have very similar aspects. And in the paper that I'm going to reference at the end that we wrote up, we talk in more detail about all the methods. So I just wanted to think about why we're doing this. And again, I'm going to get to simulations on the next slide, but just so DC2, which is the differential expression piece, it's using this size factor, but then it's fitting a linear model, and it's fitting a negative binomial linear model, and so it just allows us to do it allows us to do hypothesis testing or parameter estimation or whatever the pieces are, again, in that apples to apples comparison. So that normalization piece then just fits really nicely into the likelihood model and whatnot. And this is a newer paper where they're actually doing this. This plot comes from this paper right here, and then these people wrote a really great package, our package, to do this. But they're fitting sort of models over time courses in such a way that we can do this parameter estimation through likelihood methods, but then we need that size factor to be able to fit these models. Okay, so there's a lot of words on this slide, and they don't really say a whole lot. They say the same thing that those four plots showed, these four. So we have these four plots, and this was our simulation. So, so when we did asymmetry, we did 75% up and 25. Oh, sorry. 75% were up, twofold upregulated under A, and 25% were fourfold upregulated in B. So you would call that downregulated in B. Asymmetry with different mRNA, we did twofold and twofold, right? Because twofold and twofold would give us a different amount of mRNA. So these are just sort of the simulation details, and they just equal this slide, which I think is a little bit easier to think about. Okay, so, right, we keep using this word differentially expressed across conditions A and B if there's a difference in the amount of mRNA per cell. And really what we want to know about is differential expression in the population. That's the whole point of what we're doing, but what we have are samples. So we measure differential expression in the sample. So that's, welcome to statistics. This is all of statistics in a nutshell. You want to say something about a larger population, but really what you have is your sample to work with. And so we have to kind of assess what's going on. We know the truth, we know the population because we're simulating it, and we get these data sets. And so we're going to use the false discovery rate to assess what's going on. And so the false discovery rate is going to be the proportion of statistically significant genes which are not differentially expressed in the population. And we generate 1,000 genes, these two conditions, A and B, 5 samples in each. For all of the simulations, I'm going to use DE-seq hypothesis testing, which means a negative binomial model, sort of that I just showed you, but every one of them is going to have a different normalization, like that S size factor is going to be different. So here's our first one, symmetric, same amount of mRNA per cell, and you see that they're actually not that different. So, the y-axis here is the false discovery rate, and the x-axis is the total proportion of differential expression. So what I mean by that is, when I said that 50%, 50%, I didn't mean that 100% of genes were differentially expressed, right? So if I come along here to my x-axis, and I say 50%, that means that what I have is 25% up, 50% not differentially expressed, and 25% down. So that 75, 25%, 50, 50% was a percent of the percent of differentially expressed genes. I don't know if that makes sense, but anyway, by the end here, now we have 100% of our genes are differentially expressed, so we should have 0 false discoveries. Guess we could get them false. We could get them false if we got them the wrong way. But anyway, they're all differentially expressed. So anyway, they all do pretty well. You see a couple of flips here and there, but okay. So now we've got asymmetry, but we have the same amount of mRNA per cell, and then you start to see some kind of weird things. So you see these false discovery rates that are going. I mean, granted, these are pretty high proportion of differentially expressed genes, but DE-seq sort of requires that median, right? It's kind of thinking about what's happening with the median. So all of a sudden, if you're at 75% of your genes are differentially expressed, then the median becomes a lot harder to figure out. And you can see here that your false discovery rate. So let's see what the y-axis there went to 12%. So see the y-axis goes, so we're talking like 10%-ish and lower, whereas the y-axis here goes to 40% of false discovery rate. So now we've got this condition where we have symmetry, we've got the same number, but that we have this different amount of mRNA per cell, and DE-seq does just fine, right? But now all of a sudden total count is out of whack because we've got the different mRNA per cell, and that's when it falls apart. Right, as expected, given what we know about sort of what goes into the methods. And last, we have asymmetric with a different amount of mRNA per cell, and as expected, that it's harder. This is a harder problem, so we do worse. Okay, and this is just those same 4 plots all together in kind of a random order. I'm recognizing. So really the key takeaway here is that a lot of these methods that use sort of that middle to benchmark, right? That middle to kind of grab onto, they fall apart when you have a lot of differential expression. We also did validation, or I wouldn't really call it simulation, but we did some work on the C and MAC-C data, where the genes had been PCR validated, right? So we had the truth. They were called either non-differentially expressed, no call, or differentially expressed. There's no real reason to believe that there's systematic differences in the mRNA, but we didn't have any way to measure that. And there were approximately the same number of up and down regulated genes in the analysis. I don't know if something wonky happened, but and what we see when we look at the ROC curve is that most of the methods are doing pretty similarly. Poisson-Seq that I didn't really talk about isn't quite as good. And total count has no problem here, right? Total count, that sort of RPKM, has no trouble getting the right answer because, again, we're assuming that the genes are that the mRNA is not particularly different across the two. So we also, I don't know if I wrote this on here. Yeah, I don't have a slide for it, but we also then, and I can't remember which way we removed it, but we took out like the majority of the upregulated genes or something like that, right? So that there would be an asymmetry to what was going on. And now all of a sudden, when there's this asymmetry, the methods like DESeq, right, that's the black line is the DESeq line, don't do as well as total count in finding the different expressions. So, what are my sort of takeaways from this? Well, if you want to know difference in the transcriptome, then the library size normalization is the perfect choice, right? But that's what you're doing. You're finding the rate across that entire library size. So that's just really important for you to know. If you've got them, use them. Like if you have spiking controls, those are often the best way to go about this problem because then you don't have to worry about all the other aspects of what's going on. And RUV that I didn't talk about tries to address some of this. It's a newer method. But the key thing is that the correct normalization method totally depends on assumptions that are valid for your biological experiment. That I can't tell you which one is the best. And that incorrect normalization can lead to problems in downstream analysis. What I've shown you here is inflated false positives. But again, depending on what you're doing, if you're doing some kind of like clustering or whatever it is, that getting these numbers right is really important for it's the first step. You need to have the correct measurements to do whatever kind of downstream analysis you're planning. And I've given you kind of these extreme examples, but they do exist, right? So kind of again thinking about what you're measuring and whether your experiment is if you're seeing global changes or not. And it's just like voting, right? There's no perfect voting method that beats all of them. There's no perfect normalization method because you can always find cases where the assumptions are violated. And there's a paper that we wrote on this that has a lot more details of the methods in particular and the assumptions and whatnot. So it has all those same pictures. That was just brilliant of Kieran. Like he would bring in his work and he would be telling me about this and be like, can you draw a picture? Can we talk about pictures? And then he would come up with these pictures. And I was like, now it's clear. It's clear with pictures. Anyway, thank you.
So one thing that I work a bit with RNA-Seq, and one thing that is very hard to do is to correct for cell counts. So because you're saying that you want to know how much difference there is in RNA per cell.
Yes,
right?
That's right.
If you have different cell proportions between the two experiments.
That's right.
Then how do you do that?
Well, I mean, I feel like that's sort of what we're trying to get at here, right? I didn't actually dig deep into that. But the point is, you might have more. I mean, that would be like on the very first slide, right? You just might have more cell counts, but you want to know per cell, and so then the rest of it follows, right? So somehow kind of setting, this gene has an mRNA expression per cell, and it's the same as this one. So divide those two and then divide everything by those values.
Right. Okay. But and then you would say that you're looking at sort of the stereotypical cell in that.
Yes.
In that mixture.
That's exactly right.
Because if you're looking at blood or basically any tissue, you will always have multiple cells that will have multiple levels of mRNA. So
And then then you've gone like on that's another level. You're totally right about that. That if you've got like mixed cells, then figuring out well then you're like asking a question about how do I take this sample and break down the proportion of different types of cells, right? And then the proportion of mRNA in each of those types of cells, and that's it. That's a whole new level, and this does not address that at all. But my guess is that these ideas could easily play into that, right? Like asking yourself, what is it that I'm trying to measure, and what happens when I measure it wrong, and what happens when the proportions are wrong? Like I think a lot of these ideas could really inform that indirectly. Yeah.
What about the EQTL studies? Does the normalization matter for the EQTL studies?
I don't know a lot about it.
A bit.
Yeah.
So, at least in EQTL studies, I know that people kind of have given up on this problem. And what they do is they just quantile, they have big samples and they just quantile normalize everything.
Yeah.
Okay. So,
Well, that's another thing people do here is they set the 75th and 25th. Is that what you mean?
well, yeah, but they have like hundreds of samples or
Yeah.
thousands, so that way they can basically convert everything to the quantile, right? And if you do that, do you have similar artifacts or?
Well, I mean, that's sort of what we're doing here with the median, and some of these other methods do it with different quantiles. So let's see what this is. Yeah, see, the quantile, upper quantile, and median are all just, they're slight deviations because they all do sort of a different, reference sample or this or that or the other thing, but like that, yeah, that second row there is all trying to use, saying like, oh, the quantile is sort of better than the median for this reason or that reason, and I think it depends on what proportion of genes are upregulated.
So another area that people are applying these types of methodologies is when they look at other types of sequencing, like CHIP-seq, measure cell modifications or chromatin capture assays. Has there been any work translating these findings towards that field?
I haven't seen that, but you're right that like all the ideas of what are you measuring? Where does the variability come from? It's all like it can be translated. It just needs to be done with somebody who has the expertise in the technology and whatnot, with somebody who has the expertise in the mathematics. But yeah, I'm not aware of it. Yeah.
So also, you have specific thoughts on in terms of experiment design. So how does this inform what you would want? How do you want to set up the experiment?
I mean, I don't. I guess if you can have spike-ins and whatnot, that's related to experimental design. But I don't. I think it's the opposite way. I don't think that this informs your experiment. I think your experiment informs this. So my collaborator, he works with a protein called RPOS, which is a global regulator in E. coli. And so when he's able to experimentally sort of turn it off at different amounts, so he can turn off the RPOS, partly or like mostly or none or all or whatever. Because it's a global regulator, it's going to change like the vast majority of things, right? So See, his experiment is what he wants to know biologically. So he tells me about what the expectations are, and then I translate that into, therefore I need to make sure I'm normalizing correctly. I wouldn't go the opposite way. Like I would never tell him what experiment to run. You know what I mean? I mean, but always more samples are better, if that's what you're asking. More samples are better. All right, thank you.